Hello, everyone, and welcome. Thank you for joining us for today's session, the fifth in our new webinar series titled Environmental Monitoring Considerations for Gene-Drive Modified Mosquitoes. My name is Justin Overcash, and I'm with the Foundation for the National Institutes of Health, where I work as part of the Gene Convene Global Collaborative. Our mission is to support and advance best practices and informed decision-making for the development of genetic biocontrol technologies to improve public health. To do this, we work to identify and address key questions, provide technical advice, and strengthen capacity and information sharing. An important part of the testing pathway for new genetic vector tools will be post-release monitoring for safety and efficacy. This webinar series will highlight current vector and biodiversity monitoring efforts across Africa and introduce potential methodologies and tools that could be used to support environmental safety monitoring following release of gene-drive modified mosquitoes, which are being developed for malaria control. Sessions will take place weekly through the first week of June. Before we get started, we have a few housekeeping reminders. Please email events@fnih.org if you have any technical issues. Your video and audio will be off during the webinar. The presentation will run between 45 and 60 minutes. Feel free to submit questions anytime in the chat. Questions will be addressed at the conclusion of the presentation. All webinar recordings will be posted on the Gene Convene Virtual Institute and the Gene Convene Virtual Institute YouTube channel. Today's webinar is titled Large-Scale Assessment of Insects Using Targeted Sequence Approaches, Opportunities for Gene-Drive Monitoring, and we are honored to be joined by Dr. Mara Lonigzak. Dr. Mara Lonigzak is an evolutionary geneticist whose work bridges fundamental research in biodiversity with impactful contributions to global health, particularly malaria control. She leads cutting-edge efforts to build genomic resources that both deepen our understanding of evolution and enable more effective vector control strategies. Dr. Lonigzak earned her PhD in population biology from the University of California, Davis, where she investigated sexual conflict and evolutionary dynamics in Drosophila. She continued this line of research as a postdoctoral fellow at the University College London using transgenic approaches to explore the functional roles of seminal fluid proteins. After a brief hiatus from research, she shifted her focus to malaria vectors during a postdoc at Imperial College London. In 2012, she was awarded a prestigious MRC Career Development Fellowship and subsequently established her own research group at the Wellcome Sanger Institute in 2014. Since then, Dr. Lonigzak has led several major initiatives, including the Malaria Cell Atlas, the Anopheles Phenestis Population Genomics Project, the Anopheles Reference Genomes Project, the ANOSPP Project, and the BIOSCAN UK initiative. In recognition of her leadership and scientific contributions, she was named a hero in the field by Bill Gates in 2023. Before we get started, a quick reminder, I will be moderating a Q&A session following the presentation, so please feel free to post your questions in the Zoom chat at any time. Additionally, today's webinar is being recorded and will be posted to the Gene Convene YouTube channel after the event. With that, I'll turn it over to our speaker, Dr. Lonigzak. If you can go ahead and share your slides, we can get started.
Thanks, Justin. Okay. So it's a real pleasure to be here. I'm going to talk about two projects that we've been doing for the last few years, doing this sort of work, large-scale assessment of insects using targeted sequencing approaches. And then at the end, I'll talk a little bit about some early work we're doing to establish a gene drive panel, gene drive monitoring panel. Okay, so these two projects we call one BioScan and one ANOSPP. And at the Sanger Institute, we're core funded by Wellcome. And so we have to come up with kind of big, large-scale, long-term projects. And so these are two that I put into our last funding proposal sort of 5-6 years ago. And BioScan ambition is to, it's very inspired by an international effort to ramp up the scale of species discovery and understanding through the International Barcode of Life. And our specific UK-focused project is all around trying to gain a handle on spatiotemporal changes in diversity of insects. ANOS project is focused really just on Anopheles mosquitoes. So it's not looking at broader insect diversity, but Anopheles themselves have 500 described species. So there's a lot of diversity within the genus. And ANOSPP was designed to really try to understand that diversity in systematic and standardized ways. And most of what I will show you today is focused on Africa. Okay, so at the Sanger, we have a core sequencing facility, and so we are in a great position to do things at very high scale and to try to drive costs down. We're not super great at speed, and that's a little bit of, the way we operate is kind of, it tends to be sort of slow because we have lots of time to develop and develop the projects and analyze the results. I think if we think about in-country monitoring of either anophelines or kind of impacts on other insects in the region or other species in the region where gene drive might happen and be released, speed actually probably is going to be more important than scale. And so the cost really needs to stay as low as possible. The speed, and we need to have rapid turnaround. So with these things in mind, recently in the last kind of year or so in my team, we've been trying to wean ourselves off these very expensive, high throughput machines. This Revio that we use for BioScan costs $500,000 just for the machine, and move on to these sort of benchtop cheap sequencers that can still do an awful lot of sequencing for very cheap, but don't have that kind of big upfront capital expenditure. Along with, I think, and I want to emphasize this, most of these more expensive machines that people use to sequence also have very high cost annual service contracts, which people sort of overlook often when they're thinking about how do you sustain sequencing in country. So I think irrespective of kind of which machine you end up going with, there is a need to have considerable and sustained investment in both people and lab equipment to do sequencing. It's not something that you can just pick up and do without having a bit of investment in it. Okay, so underpinning both of these large-scale insect projects that we do, ANOSPP and BioScan, is something that we innovated in the team around when we were trying to actually extract DNA from museum-preserved mosquitoes, because the museums did not want us to grind up the mosquitoes, but we wanted to look back in time at sort of how genomes have been evolving over time. So the museums were willing to let us use the mosquitoes to take out the DNA non-destructively, but not destructively. So this has actually reshaped how we do all of our modern work. And we developed this very cheap buffer that basically you just add on top of an insect and it leaches out the DNA and leaves the specimen intact. And the costs associated with this are really around the plastics primarily. It's a pretty straightforward protocol. We work in plates because, again, we're operating at scale. And I think for any kind of monitoring, plates are a much better approach than single specimens in single Eppendorf tubes. So we basically provide these plates to all of our partners. They're already pre-filled with ethanol and they have strip caps on them. And people just take the strip caps off and one by one put an insect in each well. So I think also in thinking about kind of moving this to a sort of basic lab where the infrastructure is minimal, there are these plate-based robots that are benchtop and pretty cheap that can move liquids on and off all on a whole plate at one time. So I just wanted to mention that because I think it's worth thinking about, like, in terms of, we're not only thinking about kind of like the technical approaches, but also the cost associated with running these technical approaches in country. So effectively, this whole protocol works by the mosquitoes or the insects are kept in ethanol, room temperature. For long periods of time, the DNA is preserved just fine, and the specimens are largely preserved pretty much intact. We currently, the way it works is we then receive the plates back at Sanger for both projects at scale, and we then remove the ethanol, add on 100 microliters of this lysis C buffer, and the protocol, you can find it at that tiny URL if you're interested. It's very cheap, non-destructive DNA extraction buffer. And then we leave those overnight incubating at a slightly warmer temperature. And then the next day we remove all that DNA in the lysis C now, move that into a fresh plate and put ethanol back onto the specimen plates. You also don't have to do that. You can leave the DNA sitting in with the specimen and it continues to extract over time, and we've found that the DNA is still viable to use for sequencing, years later, even if the insect is still in there with it. So this has driven down, in order to operate at the kind of scale we're trying to operate at, we can't have extractions costing us £1 or more, which is what they typically cost when you use a kit. Per specimen, it would have been 1 million pounds just for DNA extractions for BioScan, which was not feasible for us. Also underpinning both of these projects, and I can't emphasize this enough because I think it's a big problem in the field, is standardized and comprehensively recorded metadata. And by that, I mean like people need to be using the same terms to mean the same things, and they need to be using the same versions of the same words. So it's really hard when you're receiving samples from all over the place, when people have categorized, for example, female as F or lowercase f or FEM or, there's a million different variations on how to categorize things. And so one of the things we've really worked hard on is developing what we call our metadata manifest. And this actually goes through a process of, so our partners are largely either interested in the work for their own right, they're research scientists on their own, and they want the data back for their own research, or they are interested in the results. They don't actually want to look at the data, but they want to know what species are present in their region. So they have to fill this manifest in, and it's a pain, and it's really prone to typos. So if I can get out of my, I just wanted to do a quick demo to show how we've made a drag and drop portal here where people can take the manifest. So this is a manifest, for example. And you can see it's detailed. It's got a lot of, every row is data on a single specimen. And sometimes people don't know anything about the
Hey, Mara.
Yes?
Are you sharing your screen? Because I can't see
Oh, did it go out?
potentially.
Okay, let me try again.
There we go.
Okay, sorry about that. Thanks for telling me. So this is what a manifest looks like. So this is telling us the name on the plate. So this plate was called Camp 129, and this is the well. And this is telling us what's in that well, a whole organism, and what solution is in the well, et cetera. So it's all standardized, standard GPS coordinates, et cetera. People will not necessarily know what species they're putting in there, so they leave some of this stuff empty. But the eagle-eyed among you might have seen a little bit of a typo here where somebody has put in banana instead of what they needed to put in. And so what we have is this portal, because we're working with so many partners and people make lots of typos, you can then drag and drop your manifest into this portal. And so that thing I just showed you is there, and it tells you where, so I'm going to hide this panel, where there's invalid values. So this kind of process of standardizing metadata has been something we've worked a lot on, and I think that there's still room for improvement there in terms of getting like apps that basically do that for you as you're typing stuff in. Okay, I'm going to try to go back to the presentation now. Can you still see my screen?
Yes, we can see the presentation now.
Okay, great. Okay, so that's the background on kind of the advances we've made on DNA extraction and on metadata that really have been important for both of these, for scaling up both of these projects. And now I'm going to tell you about BioScan. So BioScan is really just using very simple DNA barcoding. It's one mitochondrial marker. It's 658 bases long in most insects. And it acts very much like a product barcode. So it's got very highly conserved primer binding sites, and in between it's got something that's kind of like a barcode or a fingerprint that's pretty unique at a species level. So it's, and there's a huge amount of global work on this. So there's a giant database called BOLD where you can see hundreds of thousands of barcodes attached to species names especially for insects, but for actually there's barcoding done work in fungi and in plants as well at scale. So at Sanger, we have this BioScan project, which is very inspired by an international barcode of life BioScan effort to scale up species discovery. But barcoding is very cheap. It's just a little bit of data per species. You're not really trying to sequence much in the genome. You're not trying to understand anything about the genome. You're just trying to get a name on that or an identifier on that species. So sometimes you get a barcode, you can't say what it is, but you can say it's this cluster and it's unique to something else. And you can also place it phylogenetically. So you can say, for example, I don't know what species it is, but I know what genus it is. I see this BioScan and International Barcode of Life as very complementary to another global endeavor called the Earth Biogenome Project, which is working to scale up reference genome sequences for all of described eukaryotic diversity. This is kind of a one specimen per species, get the full chromosomal view on that organism. And the ambition is to do all 1.7 or so million described eukaryotic species. Of course, this is a much more expensive undertaking, but at Sanger, we are involved really heavily in this, leading the Darwin Tree of Life project, which is generating reference genomes for the species that are described in the UK. So between these kinds of projects, I think we're creating a comprehensive DNA-based library of all life. Okay, so what is BioScan in the UK? It's a volunteer model, so we operate by putting out posts to say who wants to participate. We don't pay people to participate. We send them everything they need, but it's their time and their investment in running these traps. They're called malaise traps. They look like tents and they passively collect insects at scale. And we want this to act very much like weather stations. So the way I designed the project was I wanted pulses across the country, 24 hours every month for multiple years from 100 sites across the country, so that we really get a handle on kind of spatiotemporal change, both locally, but also across the country. So our volunteers run the trap, and then they plate out every specimen that they caught into a well of a plate. And then, as I showed you, we receive those plates back. We extract the DNA non-destructively. We image all of the insects, so they look much better than this. This is just a picture with a phone. And then we sequence this small barcode to tell us what species every specimen is. And I'll do a demo in a little bit. Our data is presented via the biodiversity report card. As soon as we get the sequence data QC'd through, we put everything back up so our partners can download everything and see what species are present at their site. And as I said, the ambition is to do 1 million arthropods over the course of this project and create a biobank of 1 million DNAs for further exploration, because we only use 0.1% of the DNA for this part of the project. Here's a couple pictures of malaise traps out in the wild, and here's a map of the UK, and every blue dot is where we have a partner running a trap every month. This is what a catch might look like, and this is what a plate might look like. This is a real partner plated out their catch and sent a picture. You know, every partner does this too. They take pictures of the catch and the plate, so we have vouchers of what's been done. Robots, so we've built this robot. We are actually on our V2. This is the V1. It scans the plates from the top and takes a picture above from each well, so that we have a record of what that insect looked like. And then we work at very tiny volumes. So this is what's called, hilariously, the mosquito. And this robot actually takes 0.1 microliter out of each of those DNA extraction plates and moves it into a 384 well plate for PCR. So you can see these are very strange pipette tips that come out on a strip, and then they move this tiny volume into the plate, and they move along the strip. So it's a 2-day extraction process that's again done on robots here, because we're doing 48 plates a week, I think, doing DNA extractions, and then the PCRs that follow that. It's a 2-step PCR, which I'll explain a little bit later in the ANOSPP part, because it's very much the same strategy. And 2 steps are really good because initially you just amplify the target that you're after, but that the PCR primers that have the target primer have tails on them that allow you to then add UMIs, unique molecular indexes, or confusingly also called barcodes, DNA-based sequences that uniquely identify the specimen. So ultimately, you pool all of the PCR together from across however many plates you're working on. So for BioScan, we actually pool 96 well plates. So that's 9,216 specimens worth of data that we pool and do one library for. And then we deplex based on the sequencing tags that are attached to the to every molecule back to the specimen, the well that it actually came from. Here's a few pictures of kind of what the real images look like from our scanning camera. And you know, it's amazing. There's a wide diversity of insects and of course of sizes. And then we put all these images and all the sequence data up on BOLD, which is the Global Biodiversity Barcode of Life Index database. Sorry, Barcode of Life database. That's what it stands for. As I said, it's a non-destructive extraction approach. the vast majority of specimens look like this. There are some problems with certain groups where things do actually change their morphology and scales fall off. So it depends a little bit on the organism in terms of how taxonomists would find the post-extraction specimens as with respect to whether they can actually find the key characters. Okay, and then I think I will just again try to do this. I'm going to stop share again because it seems like I have to, and then start share again. So this same portal, which is, can you see my screen?
We sure can.
Okay. Is talking about our project. So we've had 307,000 specimens come on site. And again, this is that map I showed you. Here's all the people who are involved in the project on a partnership level. And this is the kind of protocol I've just shown you and the manifest validation portal I showed you. And then if you go to report card, you can actually see if you're a particular partner, you can select who you are, and then the sunburst here that tells you what the species are that we found so far, which is interactive. So we've had 122,000 Diptera, and it goes out in taxonomy. So this is the family, the genus, and the species level. And you can kind of click on a particular, if you're interested more in Hymenoptera, you can click on that, and then it will show you all of the different families we've found in Hymenoptera and all the different species. And the graph down below changes according to what you've selected. So I'll go back to the full one. You can see we did a few historic samples, and then we started our pilot in 2021, and then we really ramped up with partners in 2023. So we were getting in the summer months on average around 12,000 specimens coming in each month. And it slows down in the winter as people are running their traps, but of course there's not much out there. And we're in the middle of sequencing 2025's, the winter of 2024 and into 2025 now. And this is where people can come and download their data. It gives you the barcode, and it gives you, again, all that metadata that people so carefully enter is now here for them, connected to what the species identification was, or what the genus was if we can't put a species name on it. Okay, so I'm going to go back to Keynote now. And you can still see my screen, right?
Yep, we can see your presentation.
Okay, so the DNA biobank that we have for all of these specimens, for both the Anopheles project that I'll tell you about next and for BioScan, is really useful for other studies. So we are only using just a tiny fraction of it for the barcoding of cytochrome oxidase 1. We have ideas. We haven't actually done anything in this space of looking at other DNA barcode targets. So for example, what bacteria do those insects carry, or what fungi are they infected with, or what plants have they pollinated, or what vertebrates have they fed on? You can see signal in all these insect extracts of the things that the insects have interacted with. So there is a lot of potential to go back to these DNA biobanks and start to explore other species to get at species interactions. Sometimes we get those interactions for free if the CO1 happens to work in the other specimens that the insect has interacted with. For example, we've got, I think, nearly 1,000 specimens now where we can see nematodes, CO1 of the nematodes that are infecting those insects. And this DNA, we've done a lot of short-read whole genome sequencing on DNA that has been extracted this way and used for first amplicon sequencing. We go back for various reasons, and we say, actually, this is a specimen we're interested in doing whole genome sequencing on. And that's actually kind of a lot of what we do in my team, is a lot of population genomics, but I'm not talking about that today. We've also kind of vetted that we can get longer molecules from this. So these, the average length that we're seeing is kind of 10 to 20,000 bases coming out of this non-destructive approach, which means that we've also been able to generate long-read chromosomal genomes from this non-destructively extracted DNA. But that's really only worked with amplification so far, which is not ideal for creating reference genomes. So for long-read population genomics, I think the DNA is great. For reference genomes, I think we've got, there are better approaches out there to get the good DNA out. So we've sequenced 170,000 so far. Malaise traps are famous for catching mostly Diptera, so it is a sort of biased sample set, but we have a lot of Diptera diversity. The barcode of life database has a classification scheme called BINS, which stands for barcode identification number. But you can think of BINS as kind of proxies for species because not all CO1 sequences actually, people haven't, for all of this diversity that's out there, that doesn't all have a name attached to that sequence yet. And so we can talk about BINS more easily than we can talk about species. And we've detected over 9,000 of those so far in our project. The estimate on the UK species inventory is that there's something around 25,000 species of arthropods in the UK. So we've detected a good chunk of them, and many of them are observed only once. So these are rarer species. And then there are a number of bins that we observe a lot of times, more than 50. It can be at one site, but it can be across the sites. And then there's a few species that we see all the time. We've seen these species more than 2,000 times. So the main reason that we're doing this project is to reveal new species and help us monitor how species diversity changes over space and time. Many of our project partners are doing things like rewilding efforts. They want to understand our efforts to kind of change the landscape here in a framework of trying to go towards a more natural landscape working. Are we increasing the diversity of species that we encounter in the region? Insects are, of course, as everyone knows, critical to the health of our planet, and we really don't have good ways of monitoring that. I believe that DNA is a great way to monitor this. I'm excited about things like AirDNA and eDNA to work in this space in terms of, I think one of the challenges, of course, is you don't really want to be killing the things that you're monitoring. And so but I think in order to really understand things like AirDNA, what are we actually sequencing, we do need better reference indexes. And I think single specimen based monitoring for right now will help us scale up getting a better reference index, matching each of those species to the sequence. Yeah, so, and then I think these are good resources to understand the impact of climate change and of conservation efforts. And I think as relates to this seminar, to Gene Drive on insect biodiversity, the resources are limited, and so we really want to think about how do we do this in a way that actually enables a true understanding of impact. And I think that DNA is a major player in that. Okay, I'm going to switch now to ANOSPP, and many of you will know about the invasion of Stephen's eye, and I do wonder if we had sort of more agnostic approaches towards species identification for Anopheles, which actually kind of all look alike, whether we wouldn't have seen this much earlier on. These morphological and PCR-based diagnostics that are so common in the field, they are very specific. And so you look for a particular band size, you run a specific primer set because of what you think it's likely to be, and you look for a specific band size. And if you get a failure, those failures by practicality are often ignored because maybe it was just bad DNA. But ANOSPP is sort of set out to try to remedy this problem. The species diversity, as I mentioned, for Anopheles is very complex. There's over 500 species, and it's a mix of vectors and non-vectors. And in Africa, the bulk of malaria transmission is through 4 species. 3 of those are in the Gambi complex, and 1 is in the Phonestis group. But each of those groups contains about 10 species that are a mix of vectors and non-vectors. And then there's another 15 or so species across Africa that are really locally important vectors that are largely ignored. So how do we kind of create a diagnostic that will help us not make these very targeted assessments of what species we're looking at, that we actually kind of apply one tool to understand everything at the same time? I think I didn't say morphological species ID is also actually, it's not only difficult to get to species level, but it's also laborious. You have to look at every specimen individually, and the expertise is becoming rarer. And so that kind of barcoding approach I just talked about with CO1, while it works for the vast majority of insects, it actually doesn't work so well for things that are in a species complex. So CO1 alone, even though, if we think about the bin I just talked about, Gambi, arabiensis, and Kalutzi would all be in a sort of suite of smeared bins. They have a number of bins, but they are actually, they're not faithful to the species. And that's because there's still ongoing interbreeding occasionally between those species. So mitochondria have moved between them. So you can't trust the CO1 to faithfully tell you what the species is. You really need to look at the nuclear genome. So how can we create a kind of common language approach like DNA barcoding, but that works for any anopheline? And that's what's really motivated building ANOSPP. The genus of Anopheles is very old. I mean, this genus has been around since continents that are no longer touching were touching. About 50 of the 500 transmit human malaria. And what's been really key towards developing this panel has been the access, the real privileged access to reference genomes from an early date. So we've had about 20 species of reference genomes for over 10 years, which has really made a lot of science possible. Reference genomes do all sorts of important things, but what we've used them for here is identifying those places in the genome where we can design those conserved primer binding sites that work across the entire genus, but that flank informative variable DNA. So what is ANOSPP? It has 62 generic 160 or so base pair targets across the nuclear genome. So these should work in the vast majority of Anopheles. In total, you're not getting very much sequencing data for the mosquito. You're just getting about 10,000 bases of data per specimen. We also added in 2 mitochondrial loci that target the generic Plasmodium. So this tells us if we amplify DNA using this panel from a single mosquito, it will tell us, does Plasmodium get detected in this mosquito? And if so, what is the species of Plasmodium that is detected? The panel is applicable to any Anopheles kind of sample. We primarily have used it on adults. It works fine on larvae, and I'm sure it would work fine on bulk metagenomic trapping or eDNA from larval breeding sites. We haven't really applied it in those fashions yet, but there's no reason that it wouldn't work. The non-destructive extractions have been really key. It's high throughput. So at Sanger, for the last few years, we've been running 800 specimens per MySeq run, and now we're moving on to the MinION. It's low cost per specimen, so we can do this for just a pound or a dollar or two per specimen. And it's this kind of common language and diagnostic tool. And it also, so we have encountered Anopheles species that don't have names. And although we can't put a name on it, we can place it in the phylogenetic context and understand which species group it sits in. This is just a schematic to give you a sense of where these loci are. So in the middle here is the Anopheles gambiae, chromosome 2, arm 2L. And you can see that although the arm has got different names in Albuminus and Funestus, and the targets have moved around within the arm, most of them are found in most species. And we're not trying to target any specific gene here. We're not going for insecticide resistance loci or for anything meaningful. We were really looking for sort of that thing that looks like a DNA barcode. So things that are going to work with conserved primers and give us informative data. So it's a mix of exonic, intronic, and intergenic targets. Again, we use the plate-based approach, and we think our logo is a little Swiss Army knife because we think of it as like a super simple tool that does a few things in a very basic way. So it detects the malaria parasites in the mosquito, it identifies the mosquito species and places it in the phylogenetic context. It investigates at a light scale mosquito population structure when you have lots of specimens from across a wide range, and it detects the species of malaria parasite and whether it detects the species of malaria parasite. So our project is to look at 500,000 mosquitoes across Africa. It's a 2-step PCR. And what this means is that it gives a lot of flexibility. So it's one PCR reaction. So we take out that DNA and we put it into one tube for PCR. And in that tube is 64 primer pairs, 62 that target Anopheles and 2 that target Plasmodium. But attached to it is a conserved sequence that we call the adapter. And basically then the second step PCR, we add in unique 8-base sequences. One's called an I5, one's called an I7, that then tell us which well each of those molecules came from. So this is how we deplex back to specimen level. So far, this is what we've done. We've sequenced 70,000 samples. All of these have come to Sanger in a partnership model like BioScan. So people are doing their own projects and they want to know what species they have. And so they plate out all their specimens, fill in that manifest, and send us their specimens. You can't read the names here, but it doesn't really matter. We've had a huge diversity of different Anophelines come through. And the blue is kind of like the gambiae, I think, and the purple is arabiensis. But each country's project is different. So in some cases, it was very targeted after gambiae. So I don't think for example, seeing a lot of blue here in DRC doesn't mean that there's only Gambi there. There's a huge species diversity in DRC. It's just that this particular project was going after Gambi. Whereas in Madagascar, you can see that this was much more of a general, let's get every Anopheline we can through different trap types. And there's a huge species diversity we've got through that project. You can't really see the details here, but we've created, again, kind of like for BioScan, we've created this portal where people can go and access the data. For ANOSPP, our partners send us the samples and we send them back all the DNA data, but we also send them back what we call our report card for ANOSPP. And what this is each kind of row here is a plate of 96 specimens, and each little column is a specimen, and the color gives you a sense of what the species was of that specimen. So there's a key here. So like red is Mascarensis from Madagascar, and this is Melis or Maris. I think it's Maris because it's from Madagascar. And this one here, where there's a little bit of blending of the blues and the purples, is Arabiensis. So it's got a bit more purple. There's probably a Gambi on here somewhere. I don't see one actually, where actually it's more blue. And so the Gambi arabiensis, we're working now to kind of refine our approach to differentiating those two, because we really can see the patterns clearly in this nearest neighbor approach. I'm skipping over the analytics of this because I don't think it's super important for this talk, but this is not using, the species assignment is using a Kamer-based approach matching to a reference index. It's not using alignments. Because there's a lot of indels and there's things in there that actually don't work super well for alignments. So what's also hidden here that you can't see is the name of each specimen. And if it's colored in a particular color, that tells you quickly this specimen is carrying Plasmodium, and it also tells you what Plasmodium species. So that's in this key here that you can't see super well. And also there's some kind of coding on the top of each of these panels that tells our partners how trustworthy we think the results are. So if you can see, there's a little bit of red here in this first one, and you can see there's a mix of colors, and this looks like a situation of potential contamination. So it's flagged red, saying we've seen too many alleles than we would expect if this was really just one individual. So something's gone wrong here. So this is an example sample set from the Target Malaria Initiative from Burkina Faso. They sent us I think 10 plates of specimens they'd morphologically identified as Gambi complex. They were caught either indoors or outdoors. And these had been sitting in ethanol for years, actually, in groups of 10. So they plated them out, and we didn't really know how bad the contamination would be for these things sitting in ethanol for a long time, but it seemed to be fine. We could assign them all to their species, Gambi colutia arabiensis. 2 of them actually turned out to be nili. And this is actually a very good example, of really high confidence assignments. Typically, people are not as confident in their assignments, and we find a lot more mistakes in terms of species assignment based on morphology. Because of the metadata, excuse me, that accompanied this, we could also talk about. So there's one column that talks about whether there's a blood meal present or not in that mosquito. And we could see that the parasite rate in the unfed mosquitoes was much lower than the parasite rate in those that were observed to have a blood meal. And of course, that means that those mosquitoes had fed on people that were malaria positive. It doesn't necessarily mean that the mosquitoes themselves will become transmitters. We detected 3 different species of human malaria parasite in this sample set alone, and 2 zoonotic Plasmodium. And we often detect mosquitoes that have, for example, both Falciparum and Malaria, or both Falciparum and Ovale. And I think we've even had a couple mosquitoes with all 3 species. So this is again about that metadata, which I've already kind of emphasized, so I don't need to again here. But in this particular case with this study from Burkina, they kept track of exactly what time they caught the mosquito through the night. And although there's not really a result here to declare such, it's that emphasis on having this kind of really comprehensive metadata. It allows you to then, after the fact, assign a species to every sample and say, do we see any patterns of, for example, Arabiensis starting to come later in the morning, or I think Coluzi and Arabiensis start to show up later a little bit, in more density later than perhaps Gambi. We didn't really do any stats on this. It's just a sort of display of kind of what analyses are possible when you have both the species confidently identified and the comprehensive metadata. Okay, so finally, thinking about kind of gene drive and the next steps. In my team, we're just beginning to really gain serious expertise in using this portable MinION sequencer. We've only been using it for about 6 months in a serious way, and we're just now starting to develop a gene drive panel for the MinION. To get this experience, our first strategy was to migrate our ANOSPP panel onto the MinION. We have a couple of projects with people in Africa who want to run ANOSPP locally, and so we want to enable them to do that with ONT. And this has not been a straightforward thing because, as I said, everything has been like completely engineered for extremely low volumes and robots here. So we had to kind of reverse a bunch of stuff and go back to working on a normal lab with pipettes and larger volumes. So we're still trying to figure out exactly what run times we need and kind of how much of a MinION flow cell to use for how many samples. We think that we need about 3.2 million reads per 96 well plate for ANOSPP to give us good data. So this is the same library run through Illumina and ONT, and this is that species report card. And you can see the data look excellent for ONT. So we're really able to assign species, and we think that if we can get at least 10,000 reads per specimen, then we're able to actually also see if a mosquito for any given one of those 62 targets is heterozygous. We can see both the maternal and the paternal allele there with ONT data. So we thought about originally, I mean, for years we've been talking about putting gene drive detection into ANOSPP, but ANOSPP is a complicated beast. It's got 64 primer pairs in there already. And the reality is that gene drive is really targeting a small number of species. So trying to put gene drive detection into this established panel is probably overkill. So we thought about trying to design a simpler panel that just has the targets we need, that still does all the things. It tells us about the gene drive targets and whether we can detect them or whether they are interrupted. It tells us whether there's kind of Cas9 or the guide RNA there. And it also tells us the species of the mosquito that's present. And like I said, CO1 alone won't work there. So we need to add in, we need to steal a few loci, I think, from ANOSPP or take some other ones that we'll add into this panel to make sure that we can classify to Arabiensis Kalutzi gambi. And CO1 and ITS2 will give us many of the other Anopheles species pretty well. So in collaboration with Seb and Calvin at Imperial College London, who have been really heavily involved in kind of helping us figure out all the targets and design and pull out the conserved regions of these and design the primers. We have now tested primers that work on these kind of universal gene drive cassette components of Cas9 and the guide RNA promoters that are present in the different malaria gene drive initiatives, so the 3 major initiatives, as well as their targets, so double-sex nanos, carboxypeptidase, cardinal, and miR-184. We've also added in CO1 and ITS2 for species detection, and now we're in the process of trying to add in Plasmodium detection, blood meal detection and assignment to species, and a few additional markers that'll differentiate between the species complex. So here's the sort of sizes of the primers. These are larger than what we're used to working with ANOSPP because we can do that now because ONT is a long read sequencer, whereas MiSeq, the max is 300 bases. So that's added some good flexibility in. So these are the expected target sites if there's no integration right now. So what we want to do now, so if gene drive has worked in that individual and it's now modified, these sort of target loci, the carboxypeptidase, double-sex, miR-84, nanos, cardinal would all be interrupted, and you wouldn't get any sequence data for this because the amplicon would be too big. The primers would still be there, but there'd be an insertion there, and the primers would be too big to really successfully amplify this in the context of all of these other primers. So one advantage of trying to get this done now before gene drive release is ongoing is, I think, that we can get a sense of kind of what the false positives rate what the false positive rate is. So we don't really have a great sense. For example, I don't really know what it's going to be like in terms of Cas9. Are we going to get false positives for Cas9 all over the place because there's Cas9 in the environment from different bacterial species, et cetera? Or is this going to just work nicely on wild mosquitoes? So we have this bank of 70,000 DNAs that we can go through where we know that they're wild caught. This is how natural mosquitoes should look, and we can start testing this panel on a subset of those. So we haven't really successfully run the panel all plex together yet. We're still at the very early stages of designing it. So right now we're rebalancing those primers to work nicely together, and then we'll test a variety of different templates and approaches. So as I said, we have this DNA from 70,000 wild-caught mosquitoes, and we'll evaluate sort of the target sites, and we'll look at whether they're segregating resistance already in any of those target sites. We are also acquiring from each of the three major initiatives modified mosquitoes so that we can test them individually to make sure we see the patterns we expect based on this multiplex panel. And then we're really keen to try this kind of spike-in approach where we put in, for example, 1 in 10, 1 in 100, or 1 in 1,000 positive gene-drive mosquitoes into a background of wild-type mosquitoes to see, because I think in the beginning, of course, what you don't want to be spending all that effort to look at every individual mosquito. You just want to get a sense of, is there gene drive in this population at all now? And so trying to save on cost and make sure we understand where is that sensitivity threshold. Can you actually find, using DNA sequencing, one modified mosquito in a pool of 1,000? So we can set up those all experimentally and really get a sense of how robust the panel is. Okay, so that's where we are. In summary, I think that I've talked about kind of three different things. So BioScan is really, I think, useful in thinking about gene drive and monitoring impacts of gene drive. DNA barcoding can be used at scale in individuals, like I showed, but it can also be used at scale on bulk. And this is a big area of research, it's metabarcoding or metagenomics, to monitor spatiotemporal change in species diversity. And, for example, the potential impact of gene drive on the abundance or presence of other species. ANOSPP can be used at scale to monitor the detailed changes. So, for example, if gene drive release takes place, are other species of Anopheles filling the niche? And are species changing their ranges or abundances in response to gene drive release? And then finally, this kind of marriage of the three things, a little bit of what we've got from BioScan, a little bit of what we can do with ANOSPP, and kind of adding in this gene drive to create a new panel that can be used at scale to monitor the spread of gene drive, understand what species it's found in, and also understand if there are any impacts on feeding or Plasmodium rates. So in summary, I think genomic approaches have been and continue to be key in the fight against malaria. Reference genomes have been really an important data resource for many activities, from amplicon panel design, like I showed you, to genomic surveillance, which I didn't talk about at all today, but which we've done a lot of. I think going forward, we're in the early stages of developing this and designing this gene drive detection. But I think this is when we need to be working on it, because if release is happening in the next 5 years, we want to have a really solid baseline understanding of what we observed before release has taken place. And I just want to close by saying, as new interventions roll out, we should be prepared to expect the unexpected. This is always the case. And amplicon panels really offer scale. But I think that what they don't do is they don't give you that whole genome view of what is actually going on in evolutionary responses to interventions. And so I really think, I feel strongly that some whole genome sequencing does need to continue to monitor what's going on in populations as we try to control them. So thank you for listening. And I want to thank especially these folks from my team, Mary Lou, Alex, Petra, and Francis, who have really been behind everything I showed you with ANOSPP. And Lyndall and Gemma, who have done a huge amount of work to get BioScan off the ground, and many teams and people at Sanger, and of course all of our partners across Africa and the UK who have been contributing all these specimens. And I will close there. Thank you very much.
Thank you very much. That was a fantastic presentation. We already have a few questions in the chat, but I just want to go ahead and remind everyone, if you have any questions, please go ahead and throw them in the chat. But let's get started. Our first question was from Jeremy, and this was in relation to the BioScan project that came in pretty early. His question was, are you only using Malaise trap? And I kind of had a follow-up on that, and that was, could other traps increase the diversity of species that you were identifying? Although it seemed like you were identifying a wide range of diversity just with the Malaise trap.
Yeah, so for this project, we are only using Malaise traps because they offer a very convenient way to passively, without a lot of work, you set up the trap and you go away for 24 hours, you come back and you empty the trap, collect a large number of insects. We have another kind of budding project with somebody else who's here that is interested in looking in soil at the arthropod diversity in soil, but that hasn't really, it's not up and running at scale yet. You could do this with any trap type. It doesn't really matter how you collect, but for our purposes, we really wanted to have something that was quite standardized so that we weren't adding in another variable to try to understand, spatiotemporal change.
That makes sense. Our next question was from Mike Santos. Thanks for the great presentation, Mara, and for touching on the pooled sample analysis. When can the pooling be done in the sample prep process? Or more specifically, how early after sample collection can the samples be pooled?
Yeah, so I think it really depends on how much you want to be able to go back to understand what's going on in that pool. So if you are kind of okay with the idea that you're never going to be able to really know how many specimens in that pool were positive, for example, you can pool and extract from the pool. I think so we will try that, but what we will also try, because we've developed such a cheap approach to extractions of single specimens, is actually still extracting individually, and then pooling the DNA before we run the PCRs, so that we're saving all that kind of effort and plastics and cost on PCR, but we're not saving it on the extractions, because then you can go back to specimen level and understand kind of what's your rate of positivity. I think with pooled approaches, we're really just going to be able to say something about sensitivity. We're not going to be able to say anything about numbers. So, differentiating between 10 in 1,000 and 12 in 1,000 is not going to be possible with pooled sequencing unless you can go back to the individual level. So I think, I think it depends a little bit on how, well, money. So how much, if costs are always a concern. So I sort of think that for the early days, actually, there's no real reason to try to keep that individual level. You're just trying to see where is it and is it spreading, because you can always, once you detect it, then you can say, okay, now we're going to start collecting at an individual level and starting to monitor much more finely what's going on here.
Yeah, that makes sense. Let's see, we have more questions rolling in. The next one is from Charles, and it's kind of a broad question. It just says a word about the potential of hybrid Anopheles mosquitoes. So maybe a little bit more about what you're seeing in terms of hybridization.
So ANOSPP, I think we have not seen anything that I would say can tell us conclusively that there is, for example, a Gambi-Kaluzi hybrid. There is this stuff going on in West Africa, where it doesn't actually appear to be hybrids. It appears to be kind of like another ecotype that is very closely related to Gambi and Kaluzi. And in ANOSPP, so I had a slide where you could sort of see that it was a target malaria slide where, like, I didn't actually go into the detail there because we're kind of changing this approach right now, but where with the data, we then assign each individual within a Gambi complex to its species using a variational autoencoder. And so there is a stripe in between Gambi and Kaluzi that is actually those specimens from West Africa that we think might be some of this ecotype. But that is also exactly where you would expect a hybrid to sit. And what we haven't done is gone and then like genome sequence these to figure out if it's a hybrid or if it's this kind of ecotype that's been recently published on. So I don't think it's super well powered to detect maybe F1 hybrids, but certainly not much later than that, because it's really looking at the whole data. It's looking at all the amplicons and trying to assign a species. It's not trying to assign kind of proportion. Well, actually, it does assign proportion of species, but I think that can be quite noisy with hybrids. You're seeing a bit of that background anyway. I haven't answered that super clearly. If you want to talk more, you can email me and we can talk about it.
Perfect. Let's see. Our next question from Sebald. Would any of the primers in BioScan capture the species of plants an insect has fed on? If not, have you considered including this?
Yeah, so the BioScan primer is just Cytochrome oxidase 1, and it does not capture plants. There are two other primers that originally, when we designed the project, we were going to have a panel. BioScan was going to be a panel detecting plants and fungi and Wolbachia. And but actually making that plex work has been difficult. So we've just stuck with CO1 to start the project off. I think I said earlier in the talk, the DNA is there. So you can go back, and because we've done this two-step primer design, you can just swap out the primers and everything else still kind of works. You can just add that tail, and you can do RBCL or MAC-K, which would tell you about the plants. And you can also do this, I think, at bulk. So like if you're interested in just individual interactions, you can go back to every insect specimen and amplify the plant DNA. The way that we catch insects for BioScan is in a bulk soup of ethanol. And so one of the things that happens is the pollen comes off. And so you can end up with a background soup. So you have to control for that when you're doing it to make sure that the signal you're seeing is overcoming the noise, the background noise of sort of plant DNA that's just happened to be in the liquid, or use a collection approach that doesn't have that risk of soup.
Right. Let's see. Another question from Fred. ANOSPP using nanopore in Africa is great. How transferable is the analytical pipeline for local analysis? Also for dealing with local polymorphisms and artifacts.
Yeah, so we are trying to make that all much more robust now. As I said, we're just migrating over to MinION now for ourselves, and so we've got the pipeline. It's not been trivial to get the consensus sequences built, and so I think we've made a real big leap in the last couple of months there. So I feel like ANOSPP on ONT is going to work, and I'm not too worried about it anymore. But we're just trying to kind of do more tests ourselves to get this whole pipeline robust. But it will all be accessible. And the way we'd like it to work, but I need some more support to make this happen, is kind of like how I dragged and dropped that manifest into that portal. I want to be able to support people to drag and drop their FASTQ files into a portal and basically get out the ANOSPP report card. So the QC is all happening, the species detection is all happening, and you get the report card out. But we are a long way off from that being mature. It's still conceptual at this stage, but we really have in mind kind of making everything as kind of bite-sized, modular, cheap, and straightforward to use as possible for these panels, generally for people. Yeah.
That's very cool. And I'm going to have a follow-up question on that. And that's right. You showed that slide with your kind of all of the different locations across Africa that you're getting sequence from. And obviously your partnership model is working very well just based off the number and diversity of samples. How do researchers who you aren't partnered with, but would like to partner with you, how do they go about doing that? Is there a process? Do they
Yeah,
shoot an email
we have a
out?
register your interest button on our website, and yeah, it's been an interesting thing because BioScan, we have the same button on our BioScan website, and BioScan has had a huge amount of interest. I mean, we've had so many people, and that's just one country, so many people have wanted to participate in it, and with ANOSPP, it's been much quieter. It's much more me peddling it than people coming to me saying they want it, which I think I understand it for a variety of reasons, but I also still really believe in its power. I think we're not quite there yet to make it in the hands of anyone, but PCR is in the hands of anyone, right? Like you don't have to register your interest or talk to me to get the protocol for PCR. Everybody just knows how to do PCR. And that's how I want ANOSPP to be. I want it to be, and generally Amplicon panels themselves, like not just ANOSPP, but also this gene drive panel, any panel that anybody's developing, it should be, I think that there's real value in maintaining connections and having a shared ecosystem of analysis. And I really feel strongly, it's very boring, but it's really important about the metadata standards. But I think as much as possible, it's just a tool, and people should be able to run the tool in a simple way. And so we're trying really hard to make each of the steps as simple as possible.
Since you mentioned the metadata, and I had a question on the metadata, I'm going to follow up. We have another one in the chat. I still see it. So I 100% agree with you. Right, having the data standardized is super important. I know there's a lot of talk. Right, we're entering the AI age about metadata being interpretable by AI. Are you guys thinking about right, how your data is interpretable by AI or whether AI could play a role, right? Because we're talking about huge amounts of data here.
Yeah. I mean, because we started off this project with such strict standards, I don't think we have a curation problem the way we do in a lot of other parts of our research world where we've kind of had very loose standards and it's a bit trickier to go back in time and reformat to the same standards. Um, we haven't thought a lot about AI in terms of the metadata. I think that there will be advantages in looking at the sequencing data with additional tools. I think there are nuances in there that we can see. I mean, ANOSPP, a bunch of stuff I didn't talk about. ANOSPP can detect inversions, so we can carry a type 2LA in every individual very straightforwardly, and we can discover inversions in other species too. So and you can discover some things around population structure too. So, you know, we're looking within country at different within species within country, and you can see hints of things that I think there's probably some promise in some other approaches than what we're doing, which is kind of just PCA and trees to explore some of that structure. But we haven't really gone down that road yet.
Yeah, I think 100% agree. Setting it up with rigorous standards right from the beginning really future proofs a lot of the data. Let's see. We had another question from Fred for genetically modified mosquito release projects. In the absence of multiple constructs, any single developer will be able to do their genotyping with 3 to 4 PCRs. Do you see the panel used as a first assay done or a subset of individuals only because of the cost reasons and overkill of a multiple GM panel? Let me see.
In the absence of multiple constructs. So this shouldn't, okay, I'm not sure I'm totally understanding the question, but just to be clear, with the gene drive panel that we're developing, that's one PCR that amplifies all the 3 initiatives approaches. And so we're just being agnostic right now to which approach or approaches will end up having release, and potentially the targets will change. But the strategy, I think, is that you would want a panel that's targeting what's been released as robustly as possible. So the Cas9, the guide RNA, and the target site, and anything else that's kind of added in that you want to target. It is still the one PCR. It's just a multiplex of primers. So you're not doing a separate PCR on every target and then pooling, in case that wasn't clear. I'm not sure I
Right.
answered the question.
I think you answered it, and you also hit on the fact that right whether we're talking about right for it makes sense like if you're just looking for a detection in the spread of the gene drive, having a pool of mosquitoes, you do that single PCR detect presence or absence, and then if you'd like to explore kind of a more rich or detailed look at the population, then you could move to individuals and capture it that way. So
Yeah.
I think you hit on that pretty well. Let's see, we have no other questions in the chat at the moment. I know I have another question or two. If you have a burning question that you'd like to throw in, now would be the time to do it. But while we're waiting on that, I always think, right, whether it's how you categorize your data or how you're storing your data, these are really interesting questions that I don't think get a lot of attention in these types of talks. So one of my questions was, what is the data storage requirements for all the information that you're gathering, both physical and genomic, right? Because I think those pose different challenges. And another one, because you're talking about the switch between going from MySeq to MiniIon. Was there any data reconfiguration required for that process in terms of right making that switch?
I mean, this all just sort of depends on your experience. I think of amplicon data as really tiny data. I mean, it's stuff you can analyze on a laptop. So I don't, the data storage requirements are relatively small, especially after you've kind of analyzed it, because then it's just about what, I mean, you have a file that tells you the sequence of every amplicon, but you no longer have all the reads, which is where the sort of bigger files come from.
Sure.
And all the garbage in the reads, which there is a fair amount of stuff that you just don't look at all. It doesn't deplex or it doesn't have the primers in it. And your second question was around migrating from MySeq to MinION with respect to what?
Like, is the, in terms of using different sequencers, was the data one-to-one, or did you have to change your kind of your pipeline at all?
No, we did have to change the pipeline quite a lot.
Yeah.
It's not one-to-one. ONT has a totally different error mode than MySeq. And interestingly, looking at the ONT data helped us identify something we hadn't seen in the MySeq data before, where there's 3 of those 62 amplicons that actually we get much better coverage for from ONT for any given specimen. And we've always had enough coverage because we kind of heavily oversequence. We've always had enough coverage to use those amplicons, but the coverage has been low. And yeah, we're starting to see data from those. And we are, Francis and the team has been trying to figure out exactly what it is about those 3 targets that doesn't sequence well in a MySeq. And it might be some kind of, yeah, I think he's still exploring, but he thought it might be some kind of GC content.
I have one last question, and then I'll close this out. So right, you mentioned at the beginning, you mentioned the BioScan initiative, and right, you're identifying a whole bunch of species. You mentioned some novel species. One of my questions was, what's an example of an unexpected result in terms of novel species, or maybe a species that seems to be out of place?
So novel species is a really hard one to say because because to know that it's novel, you have to know that it's somebody has looked at it and doesn't know what it is. And that's not really what we've done yet. We are running these things that we're calling taxonomy-thons, where with some of the bins that we're seeing really frequently that don't have species names, we're getting a group of Diptera experts together to meet up at the Natural History Museum in London and to look at the specimens and try to put some names on them so that some of the things that are more common we actually get names on. So I don't have an exact answer for what you're asking, but I do have a really fun one, which is so like I said, sometimes you get this byproduct, CO1, for not the insect, but something else it's carrying. And so we've seen a lot of nematodes. But I also saw in the data a few tardigrades. And that was fun. So I wanted to see a little bit more about that. And the really weird thing is that from different sites, so I can't remember exactly how many, but I think it was like 3 or 4 tardigrade sequences from 3 or 4 different insects. And these were not from the same site at the same time, but they were the same species of beetle that carried this tardigrade. So I wonder what's going on there, whether the beetle has eaten the tardigrade, and so we're still seeing the tardigrade sequence, or whether tardigrades are riding on beetles.
That is pretty funny. That is interesting. Yeah, those are hardy little creatures. Very cool, though. All right, well, I see no other questions in the chat. So I just wanted to go ahead and say thank you again, Dr. Lonigzak, for that fantastic presentation, and for everyone else for joining us today and to contributing to the discussion. We hope you will join us again next week on Wednesday at 11 a.m. Eastern for our 6th session in the series, where we will be joined by Dr. Elizabeth Clare, who will be presenting on a chance to measure life on land, the promises and challenges of airborne eDNA. This is the first of 2 webinars on the potential use of eDNA for monitoring efforts. Once again, we invite you to visit the Refresh Gene Convene Virtual Institute, where you'll find new resources, tools, and publications. While you're there, please consider subscribing to our free newsletter and stay tuned about upcoming events and activities. Thank you again, and we look forward to seeing you next time.